Okay, so we're gonna go ahead to talk about the examination for lecture two. Lecture examination four basically is applied microbiology. So we will talk about the food microbiology, which I already talked about the food safety in the exam for lecture one. This we will take about poultry processing regarding the pathogen control. I will tell you in the West Virginia, it's very important. Then we will talk about immunology and talk about the pathogen control. And then we also going to talk about the virus, which is very important right now. So we will cover those in our examination four, which is final exam. And if our final exam is not accumulated, only the last part. Okay, so let's begin and talk about here is pathogen control during poultry processing. This is a whole map which is showing you boar production in the United States, which is by state, each state, and it's statistically is a million head, which is coming from 2014. You will see that all of the poultry states, which is at south, Texas, Mississippi, Alabama, and Georgia, especially Mississippi, Alabama, and Georgia, including Arkansas and North Carolina, those are five big poultry states, which are almost responsible for 70% of the boar production in the United States. Now, the obvious reason, because it's hot. Okay, so that's why it's good for so where's it good for the borer to grow. And another thing I also want to mention, if you are interested to study poultry science, then University of Arkansas, Mississippi State University, Auburn University at Alabama, University of Georgia and North Carolina State University, including University of Texas and AEM, they have a very good poultry science program, which is a leading research in the world. So just let you know about that. When we move to the north, did you notice that at West Virginia, there is about 95.3 million head? And we know West Virginia is a mining state. We do not have a big agriculture. But if you look at this map, did you see the boar production in West Virginia actually play an important role of the whole state? So that's why we want to talk about the pathogen control during the poultry processing, because it's also important for the people in West Virginia. Now, what are the pathogens we really care about during the poultry processing, during the poultry meat processing? Well, including everything. For example, norovirus, E. coli 157H7, non 157 Shigella toxin generating E. coli, Listeria monocytogenes, Clostridium botulism, Shigella, Toxoplasma, and some emerging organisms. But be careful, very important. The most two important foodborne pathogens related to the poultry meat is number one, Salmonella, number two, Campylobacter. Now, there is more research coming showing Listeria monocytogenes has been paid much attention to that. And the urinary tract infection caused E. coli also pay much attention these days. But we always think Salmonella and the Campylobacter are two most important foodborne pathogens during poultry meat processing. So we're going to talk about these two. First of all, what is Campylobacter? Campylobacter is a curved multi-rod. It's gram-negative. It's relatively fragile and very sensitive to the environmental stress, including hot, acid, alkaline, and poor nutrition environments. But be very careful. This pathogen is a microallophilic microorganism. They require very limited amount of oxygen, 3 to 5%, and 2 to 10% carbon dioxide for this bacteria to grow. Therefore, it's very ironic. This pathogen is the number one in the poultry industry, or at least the top two. Because it's microallophilic condition, this pathogen is very difficult to grow in the lab. So kind of ironic, there's a lot of research they do. For example, Campylobacter can survive on the poultry surfaces very easily, but in the ground poultry meat, they may not survive very well. There is lots of unknown. We're still going on. We are still doing lots of research about that. Now, Campylobacter could be usual in the intestinal tract area of the wild domestic animals. Okay, they can survive 2 to 4 weeks as low as temperature at 40 degrees Fahrenheit. At room temperature, they can be survived 2 to 4 months, and the infectious dose ranged from 500 to 10,000 cells. So this infectious dose is very high compared to E. coli 015-7H7. And you know, E. coli 015-7H7, sometimes one cell will cause hemorrhagic uremic syndrome. 10 cells could cause people to die. Campylobacter, this dose is very high. It can be as high as 500. Okay, now the second one, which is we pay much attention, is Salmonella. And we mentioned this in the class a little bit. This is also a gram-negative, large-shaped motile bacteria, which is non-endospore forming. It's widely found in animals in the environment, and it's a mesophile, so grow very well at body temperature, 35-37 degrees Celsius. Its optimal temperature for growth is between 35 and 37. The slow growth observed at 5 degrees Celsius, maximum growth 45-47 degrees Celsius. Be very careful about here. I will mention again. The maximum growth temperature for Salmonella is actually a little bit milder heat, 45 to 47 degrees Celsius. I will mention it again, why it's important. They can be the pH actually very wide for the growth of Salmonella. They could be between pH 4.0 and pH 9.0. The optimal 6.5 to 7.5, that's pretty much the neutral pH, so we know about that. Now, this is the top 30 serotypes in 2005 regarding the Salmonella in the poultry meats. Number 1 is Typhimurium. Number 2, Epidermis. Number 3, Newport. And number 4, Hindenburg. Those all are very important pathogens related to the poultry meat. And you can see a lot of the other subspecies. This is a new standard performance, not really new, maybe 5 years ago, but relatively new. So here is something you need to know. It's different from E. coli 015-7H7 in ground beef and the poultry and the beef products. Salmonella and the Campylobacter are not zero tolerance. And the USDA Food Safety Inspection Service, FSIS, released new Salmonella and Campylobacter performance standards as January 2015 in broiler carcasses. The maximum allowed positive for Salmonella is 7.5 and the Campylobacter is 10.4. So you can understand that if you have 100 broiler carcasses, you do the sampling. If you have 7 Salmonella, 7 carcasses tested positive for Salmonella, this plant is still fine. If you have 10 broiler carcasses tested Campylobacter, this plant is still okay. So I want to tell you one thing, this standard performance standards is not really zero tolerance. So you need to know it's a different from E. coli 015-7H7. They allow a low amount of Salmonella and the Campylobacter on the broiler carcasses. Now, this is the standard broiler processing in the poultry industry. So we have a grow out transport, then liver ham, live ham, immobilization, bleeding, we call it exsanguination. Then we do a scourding, we do pickling, evisceration, we do chilling, immersed chilling, salting, aging, then further process and the packaging. Now, lots of here, the processing here relate to the poultry meat microbial safety. So we want to pay much attention to that, okay? First of all is the scourding processing. Now, scoldering started back to 1st century AD. What is scoldering? Usually use heat temperature to put the birds in there. Then let it be a little bit warm, and then you can do a good defeathering because the feather is easy to take off. Now, be very careful about the scoldering. There are the three things I want to mention here. Number one, when you do the scoldering, you need to make sure the birds come in always facing the clean water. That's why we say the water flow is countercurrent. Because if you are not do countercurrent, I tell you, lots of the small poultry processors don't do that. If you do not do countercurrent, the water will be more and more dirty. And every time you cause big cross-contamination and you accumulate organic matter in the washing tank. So always remember the scoldering processing, the water flow is countercurrent. Every time when the bird's in, you're facing the water and you will prevent the cross-contamination. Second, there is two types of the scoldering. Number one called hot scoldering, which is temperature between 59 and 64 degrees Celsius. The time is 30 to 75 seconds. The second one is called soft scoldering. The temperature a little bit lower, 51, 54 degrees Celsius, 90 to 120 seconds. And be careful about the temperature. This temperature control is very important. A lot of the small poultry processors, they don't do very well the temperature control during the scoldering. They're using 45, even 47 degrees Celsius water to do scoldering because their hands are easily to be coming to put in and do the defeathering. But I tell you, it's very dangerous. If you are using 45 and 47 degrees Celsius water to do scoldering, you're actually cultivation bacteria. Why? I just mentioned Salmonella, the maximum growth temperature is between 45 to 47 degrees Celsius. So never ever do the temperature is between 45, 47, and actually is lower than 50 degrees Celsius because that temperature, your cultivation Salmonella. Second, lots of people will ask me a question. Is there any antimicrobials can be used in the scoldering processing? Very few they used. Nobody ever use it. The reason is two organic matters there. Okay, it's not going to be very effective. So scoldering makes sure two things. Number one, temperature control is important. Number two, the water flow has to be countercurrent. Now the pickles, this is you need to be very careful. This is a vent structure. You have a AEM, Syrian, and this Syrian. Okay, all these structure and the Sika actually. Sorry, Sika area. This area usually you have these left foods is in there. So for the pickles, very important is the birds has to be withdraw any of the feeding about 24 hours. You can't do it too long. You can't do it with withholding the feeding for like 48 hours because the Sika area will be very soft. When you further do the evisceration, it's very easy to broken. But also you must be withhold food for feeding for 24 hours. If you don't do 24 hours, all the foods will be left in this area. What's going to happen? The carry-on bacteria during evisceration process, they're going to be contaminated. Okay, so be careful about that. The feeds withholding is very important. About 24 hours is good. Now, usually the pickers, they hang in there. Okay, they hang in there, use stainless, and some of them spray antimicrobials, but not really. They just hang in there. Now, just be careful. The most important procedure cause Salmonella Campylobacter, the cross-contamination during the poultry meat processing is evisceration process. You can see this is a bird is not off feed for sufficient period of time. you will see all these will coming out. They contaminate the whole carcasses. That's why the withholding the feed is very important. Okay, but I tell you one thing, evisceration process, either you do mechanically or you do handful is very important. That process is easier to get cross-contamination because basically you're taking out all the intestinal material out of the body. Now, after you do the evisceration, actually, right here, it's not shown in here. Lots of the companies, they do a washing. The name of this washing process called IOBW, which calls named as inside outside bird's wash. They have those nozzle and they spray antimicrobials used in this process. This whole process is about 30 seconds to 1 minute. How many log reduction can be causing by IOBW inside outside bird's wash? It is about maybe 1 to 2 log. So 90% to 99%, not really killed everything. Be careful about that. So they do an inside outside bird's wash before they do the chilling. Now, what are the antimicrobials? They can do an inside outside bird's wash, or we call it an online reprocessing process, OLI, online reprocessing process. This is a survey conducted by Dr. Shelley McKee at Auburn University a couple of years ago. So they do the industry survey of all the antimicrobials for online reprocessing and the inside outside the birds washing. The number one is peroxyacetic acids, 40%, followed by chlorine, which is 22%, then by acidic and acidified sodium chloride, and the CPC, we mentioned in examination one, electrolyzed water and bromine. Now, the number one is peroxyacetic acid, number two is chlorine. Now, why? The reason is very easy. In the United States, lots of the poultry meats, they actually export into the other country. If you wanted to export your birds, go to Russia, they not accept anything which is treated with chlorine. That's why peroxyacetic acid is used very widely in the industry. The only reason is those birds need to export to Russia. Okay, so that's why they have to use it. And then we did a lot of research for peroxyacetic acid, PAA, which is in our lab. It's very effective. And I want to mention a little bit about chlorine. Chlorine is very easy to be degraded by organic matter in the water solutions. So, which means if you have any organic matter or dust there, if you're preparing 1,000 ppm parts per million chlorine, those organic matter and dust will decrease the effective chlorine concentration back down to below 4 to 5 ppm in like 30 seconds. So chlorine is very sensitive to the organic matter. And the second thing I want to also mention is chlorine, if you repeatedly use, it will generate chlorine off gas. And sometimes accumulated chlorine products will cause chloramine to accumulate that might not be friendly to the environment. So lots of the places people don't use chlorine, but still lots of places people like to use it because chlorine is very cheaper. It's $6 for 1 gallon. It's very, very cheaper. Okay, so just let you know about that. Now, very important, you need to know, the most effective control of pathogen during the poultry meat processing, let's back here, is actually the chilling processing. That's called immersing chilling in the United States, widely used. So after you do inside outside the bird's wash, the bird is immersed in the ice water, usually with antimicrobials. Now, what are the antimicrobials they use? The survey also showed number one is peroxyacetic acid, 35%, followed by chlorine, then some acid, then followed by very limited people use monochloramine and acidified sodium chloride, ACS. Why it's most effective? First of all, you're using antimicrobials. Second, the time for the chilling is usually very long, somewhere between 45 minutes to even maybe half a day. So the chilling process, you immerse the whole birds with chemicals, with antimicrobials. It's most effective. Okay, but I also gonna mention there is something very important. This is something, what I tell you, is the scalding temperature is important. So let's back here. You see the hot scalding temperature and the soft scalding temperature. I just mentioned, I said the temperature cannot be too low because your cultivation bacteria. But I also tell you the scalding temperature cannot be high more than 65 to 70 degrees Celsius. Why? If you are using very high scalding temperature, all the fatty acids, fats will be extracted from the surface and they will be stick on the surface. And when you go to the chilling tank, all those fat will be floated on the chilling tank surface. So if you're over scalding, what happened? Look at this chilling tank. They are all kind of fatty acids right there. And they are a good reservoir for bacteria cause cross-contamination. So there's a research showing if you're 40% positive salmonella birds coming, you do chilling process. If this is what your chilling tank looks like, because you did not control the scalding temperature very well, the fatty acids accumulated, the birds come out, you get a 53%, which is 30% more positive samples because the fatty acids accumulated on the surface cause cross-contamination. So be careful about the scalding temperature. It's very, very important. People usually don't care about that. Low is not good, high is not good. If too high, the fatty acids accumulate. So what they should do, this is exactly the scalding temperature, what they should be looks like. Can you see it? Very clean, after scalding, very fresh, and the birds right there is ready to go. This is the something which is widely used in almost all plants in the United States for the immersion chilling. That's a picture for that. Now, there's a little bit of difference. In the European countries, they're not approved to be using immersing chilling. They use air chilling. Okay, so that's air, the cold air comes back and forth. They use air chilling. Now, to be honest, from the microbial standpoint, air chilling not be very effective for control the pathogen. This is, you can see the data. This is the percentage of salmonella positive borers existing on the chiller in the European countries, in Australian and the United States. You can see that nationwide, 5% positives, carcass positive for salmonella post-chilling in the United States, very good control. In the EU country and Australian, because they are using air chilling, the positive rate is very high. In EU country, European Union is 15%. Australian is as high as more than 35%. So the United States control very well. And the Campylobacter is even better. You can see that. Post-chilling Campylobacter positive rate in United States is 10%. In European countries, 75%. Post-chilling Australian, as high, more than 80%. So you need to know our poultry meats actually in the market is pretty safe compared to the other countries because we do a good chilling job, has enough time and antimicrobials, and also we have to control the scalding temperature very well. Okay, so we're going to go back to talk about something really specific in this state. This is called the mobile poultry processing unit. Remember, we talk about 95.3 million broilers generate every year in West Virginia. They are not by a large industry. Most of them is a family-based business, and the Department of Agriculture at West Virginia started to introduce the mobile poultry processing unit and co-worker with Farmers Market Association. So I'll give you some example. This is open air MPP or mobile poultry processing unit. This cost is about $30,000. You can do 90 borers per hour and on farm 50 to 300 birds per batch. But what the bad thing you need to be careful. Lots of the state, including Kentucky, they try to take out of the poultry industry. The reason is the waste is very difficult to control. So the same thing here for MPPU. Not all the wastewater can be captured. And also surface cleaning is very important. We mentioned earlier, if the surface cleaning is not do very well, what happens? The bacterial biofilm will be accumulated and very difficult to take it out. So that's why I want to tell you one thing. Open air mobile poultry processing unit, the wastewater is difficult to capture and the surface cleaning is difficult. But the good thing, it's very cheaper. This is the enclosed MPPU, which has been introduced by our department, Department of Agriculture at West Virginia. It's very expensive, $90,000, the whole facility. You can treat 120 birds per hour. Per batch is 200 birds. And lots of them is USDA FSIS inspected facility. The good thing, more surface clean, more wastewater and solid waste collection. This is something which is done at Dr. Joe Mott's lab in our facility. Now we have a very good poultry farm, poultry processing units. I did the research there. And we also do some of the demo in our local area. This is what we did for extension training at Marshall County at West Virginia. So you can see we handing on there and do the exaguization. We do a scottering, a manual scottering. We do defeathering, then we do manual evisceration, and then we do the chilling. Our chilling time is with ice water about 48 hours, and this is a trailer, and the trailer can take in all these items to most of the places in West Virginia. So just to let you know about this, what happened in here, okay? So this end our talk right now.